Welcome to the deep dive. Today we're going to do something a little different. We're grabbing a foundational source, a scientific document written under, well, some of the most extreme pressure imaginable.
And we're using it to explore the exact moment the world's most consequential pandemic threat was first identified.
Yeah, we're basically time travelers. We're putting ourselves right in Wuhan, China in late 2019, early 2020.
This deep dive is built around a scientific paper from researchers at the Wuhan Institute of Virology, or WIV. And this is the paper that first characterized the novel pathogen, which they tentatively named 2019 nCoV.
And it wasn't just a research report, was it? It was essentially the molecular warning siren for the entire globe.
It really was.
You just have to think about the speed of this work. The outbreak really starts to pick up steam around, what, December 12th, 2019?
Right. And by January 26, 2020, so just over a month later, the WIV team had already compiled and published this definitive genetic roadmap of the virus.
And that context, January 26th, that's so crucial because the numbers, even then, told a pretty terrifying story.
Oh, absolutely. I mean, at that point, China had already reported almost 2,800 lab-confirmed infections and 80 deaths.
And the scary part, it had already jumped borders.
Yeah, that's maybe the most worrying thing. It had already popped up in 10 other countries. It wasn't a local problem anymore. It was already spreading, and nobody really knew what it was.
So before we dive into the molecular detective work, let's just set the scene on the ground. What were doctors seeing that made them think this is something totally new?
Well, the clinical presentation was just severe. It was an acute respiratory syndrome. Patients were coming in with a high fever, a really intense dry cough.
Okay.
And then it would rapidly progress to breathing difficulties, dyspnea, which then just turned into severe viral pneumonia.
And what made this different from, say, a really bad flu season or just typical winter pneumonia?
Because the speed. The
Yeah.
speed and the uniformity of how it progressed towards respiratory failure.
So everyone was following the same dangerous path.
Exactly. Chest scans showed this severe pneumonia, and crucially, antibiotics weren't doing anything.
Right, so that immediately rules out a bacterial infection.
It points directly toward a novel virus. The clinicians knew right then they were fighting something they had never, ever seen before.
Okay, so the doctors know they have a highly infectious virus on their hands. Where does the WIV lab even begin their hunt once they get those first patient samples?
They started where history told them to look. They got samples, things like lung fluid, blood, from 7 patients with this severe pneumonia.
And 6 of them had a connection to the Huanan seafood market. What?
6 of the initial severe cases, yeah. And that connection was strong for the start of the cluster. But, by the time the scientists were analyzing this data, it was already clear.
It was spreading human to human.
Rapidly.
So the mission is to find the culprit. How do you go from any possible virus in the world to narrowing it down to one specific family?
Well, you use what you've learned. They fell back on their experience with the 2003 SARS outbreak, which also started in a market in winter. So they didn't start blind.
They had a lead.
A strong one. They started with a targeted search using what are called pan-CoV PCR primers.
Can you break that down for us? What's a pan-CoV primer?
Sure. Think of it like a generic search warrant for the entire coronavirus family.
Okay.
PCR primers are tiny snippets of DNA that only stick to specific genetic targets. A pan-CoV primer is designed to find and stick to regions that are common across all known coronaviruses.
So if it's a coronavirus, any coronavirus, this test will light up.
And it did. 5 of the 7 initial patient samples lit up. That was the moment they knew for sure. Yes, this is definitely a coronavirus.
So they knew the family, but they needed the individual fingerprint. How did they get the full picture?
That's where the more advanced tech comes in. They took one critical sample. It was bronchoalveolar lavage fluid, or BALF, from a patient they called WIV04.
And what did they do with it?
They performed a metagenomics analysis. And that's not just looking for one virus you already know. It's like casting a massive net into that fluid sample and sequencing every single piece of genetic material you find.
Human, bacteria.
Virus, anything.
Yeah.
And that deep sequencing, that's what delivered the jackpot.
The full genetic code.
Exactly. They assembled a nearly 30,000 base pair coronavirus genome. And the immediate comparison was critical. This new virus shared 79.6% of its sequence with the original SARS-CoV.
The 2003 virus.
The very same.
So let's put that number in context for you. If the 2003 SARS virus is our first cousin, 79.6% means this new virus is, it's definitely family.
Oh, yeah.
but it's distant enough to be a completely new threat.
That is a perfect analogy. It confirmed the threat was from that established family of SARS-related coronaviruses.
The ones known for jumping from animals to humans.
Right, but it was absolutely not SARS itself. It was a distinct novel entity
Right.
with, obvious pandemic potential right from the very beginning.
Here's where it gets really interesting. The scientific community already feared that this specific family of bat viruses was, the next big one.
And this genome sequence gave them the identity. But the next step, finding the origin, that's where it gets truly striking.
The bat link.
The bat link is just immense and unavoidable in this paper. Once they compared the full genome to the global database of every known virus, the result was stunning.
What was it?
The 2019 UNCODE genome was found to be 96.2% identical to a bat coronavirus, known as BAT-CoV-RATTG13.
Wait, 96.2%. So using our family analogy again, if 79% was a cousin.
96.2% is practically a sibling.
That's incredibly close. It suggests a very, very recent common ancestor.
Extremely close. And this BAT-CoV-RATTG13, it was previously found in a species of horseshoe bat, Rhinolophus affinis, down in Yunnan province.
Which is actually quite far from Wuhan.
Right. And the phylogenetic analysis confirmed it. RATTG13 is the closest known relative, forming this tight, distinct evolutionary lineage with the human virus. It was immediate, definitive evidence.
That this originated in bats.
Yes.
Okay, but if they're 96.2% identical, where does that other 3.8% of difference actually lie? Because that little sliver of genetic code is everything, right? It's what allowed this virus to make the jump.
That 3.8% is the difference between a virus just circulating in bats and, well, a virus that shuts down the world. And the major divergence point, the biggest change, was in the most important part of the virus.
The spike protein.
The spike protein.
Which is basically the key the virus uses to unlock and get inside our cells, correct?
Exactly. While the whole genome was 96.2% identical, the S gene, the part that codes for the spike, was a bit more different. Only 93.1% identical to the bat virus.
And the differences weren't random, I'm guessing?
Not at all. They were concentrated right at the business end of the protein.
The part that does the invading. What specific changes did they flag in the paper?
They highlighted a couple of key things. First, this new virus had 3 short insertions in a part of the spike called the N-terminal domain, compared to the old SARS virus.
And why does that matter so much?
Because that N-terminal domain is part of what helps the virus stick to the cell surface in the first place. Changes there, along with changes in the receptor binding motif, the very tip of the key that fits into the lock, it all suggested this virus had evolved a new, much more optimized way to interact with our cells.
It maximized its ability to infect humans.
Exactly.
Which brings us to the most critical biological question. What lock does this new key fit?
And they confirmed a terrifying link to the 2003 SARS outbreak. It uses the exact same cellular entry receptor.
Which is?
Angiotensin-converting enzyme 2, or ACE2.
ACE2, that was the mechanism that made SARS so severe almost 20 years earlier. And now here's this new sibling virus using the same highly effective doorway into our cells.
That's the critical biological discovery in the paper, and they went a step further to prove it, which is the big aha moment. They used lab cells, HeLa cells, that they engineered to have ACE2 receptors from all different kinds of animals.
So human, bat, pig.
Exactly. Human, Chinese horseshoe bats, civets, pigs, and mice.
And what happened when they tried to infect these different cells?
The virus got in. It successfully entered every cell that had ACE2 from humans, bats, civets, and pigs.
But not the mouse.
Critically, it could not use the mouse ACE2. So that experiment was definitive. ACE2 was the way in.
That's fascinating, but wait. If it can use the pig and the civet receptor too, doesn't that suggest there was probably an intermediate host? Something between the bat and humans?
It raises a vital question. And the paper doesn't name an intermediate host. That investigation was still very much ongoing.
Of course.
But the fact that it maintains this high affinity for ACE2 and other known coronavirus hosts, like civets, certainly fueled that initial search for an animal link in the market.
It showed the virus wasn't just for humans, it was a multi-tool.
A well-equipped one, yeah. They also confirmed it didn't use any other common coronavirus receptors, which helped simplify the epidemiological hunt.
And by this point, they had actually isolated the virus. They could look at it.
Yes. The WIV04 strain was successfully grown in lab cells, and when they put it under an electron microscope, you could see that classic crown-like shape, the corona, confirming its visual identity.
So with the full genome sequence, the race was on to build defenses. What diagnostics did they create right away?
They immediately developed a quantitative PCR test, a qPCR test. And for the initial target, they chose the receptor binding domain of that S gene.
The part that was the most different, that seems a little counterintuitive.
It does, but it ensures specificity. Because that part had changed the most, targeting it meant you could be absolutely sure you were detecting 2019 nCoV and not some other, milder coronavirus that was already out there.
Ah, that makes sense. You get a very specific confirmation.
Right. But they were also pragmatic. They recommended that for routine clinical testing, you should probably use other more stable targets like the RDRP or E genes, because those are less likely to mutate over time. They were already thinking about long-term reliability.
And they also developed serological tests, right, to see if patients were mounting an immune response.
Correct. They developed antibody tests, ELISAs. They even had to get a little creative and use a protein from a related bad virus as the bait to catch the human antibodies.
And did it work?
It worked perfectly. All the patients they tested showed a strong positive for viral IgG, which is the long-term antibody response. That completely cemented the link between this new virus and the severe illness they were seeing.
So this research gave the world its first clear look at the enemy. But what questions were still open when this paper was published? They couldn't have known everything.
Oh no, and they were very transparent about the gaps. The gold standard of proof in medicine is something called Koch's postulates.
Right, where you have to infect a healthy animal to prove the pathogen causes the disease.
Exactly, and that definitive verification was still pending.
So the evidence was overwhelming, but that final proof was still in progress.
Right. Plus, the precise transmission route was still technically unknown, though they proposed airborne transmission was highly likely, given how fast it was spreading.
And what was the biggest unknown?
The biggest urgent question was, will this virus continue to evolve? Will it become even more virulent? That was the thing everyone needed to watch.
So what does this all mean? We've traced this incredible story from a clinical cluster in December to genome sequencing and diagnostic development just a month later in January. I mean, the speed is it's a testament to global capability.
It's truly staggering. This one paper gave us the core sequence, the origin pointing to bats, the mechanism of entry through ACE2, all the essential information public health needed to start building countermeasures within weeks.
And of course, that name, 2019 nCoV, it didn't stick around for long.
No. Shortly after the paper was accepted, the official names came out. The virus was named SARS-CoV-2 to recognize its relationship to SARS.
And the disease it causes.
It was named COVID-19 by the WHO.
The identification was done, the race was on. But I think the most sobering part of reading this foundational document is looking at the long-term recommendations the authors made.
They recognized the cyclical threat immediately.
They weren't just thinking about the next few months.
No, they looked decades ahead. They stressed the long-term need for broad-spectrum antiviral drugs and vaccines, not just for SARS-CoV-2, but for this entire cluster of related coronaviruses. They knew this family would produce future threats.
And what was the single most vital policy change they called for? The thing the science was screaming even back then.
The paper concludes with this clear, urgent recommendation: the critical importance of implementing strict regulations against the domestication and consumption of wildlife.
And that really highlights it, doesn't it? From day one, the foundational science pointed to that human-animal interface where viruses like BATCoV, RATG13 jump hosts as the permanent fault line for future pandemics.
It remains the essential knowledge. Building on this initial discovery, that is the thing we must act on to prevent the next one.